introduce Matthew?
Thank you very much, Vincent, for the introduction. So indeed, I'm going to talk about PSEA-Quant, a protein set enrichment tool that we felt in the Yates lab was becoming a necessity at some point in order to analyze the large proteomics data that we're producing. And this was a collaboration with built with Naveen Ranyar and Daniel McClatchy that produced the data in order to build the tool PSEA-Quant. So maybe just to start with, if I can actually change the slides, okay, it works. Just to start with a little bit of motivation, labs like ours and multiple proteomics labs are well aware of the fact that most of the proteomics experiments that people generate will yield long lists of proteins that sometimes will be characterized and sometimes others. And usually with these long lists of proteins, you try to do some sort of functional characterization of some candidates that are interesting based on the type of experiments that you're performing. You might also be interested in trying to infer the function of individual proteins in your proteomics analysis, or sometimes you might be interested in looking if there are certain biological processes that are differentially expressed in that list of proteins that you've analyzed. And most of the times people will rely on analyses such as the gene ontology analysis or GO analysis, where the goal is to do a statistical assessment of the overrepresentation of a certain GO or GO term in a given data set. So basically the idea is you have a given proteomics data set and you are interested based on certain criteria, for instance, in this example, on the confidence of the identification of the protein and the abundance of the proteins. You are interested to see if there's an enrichment, let's say for these high abundance, high confidence proteins for a certain gene ontology protein set. And there are a bunch of tools that have been proposed over the years. Some of these are listed here. Some of you might be aware of David that implements also a gene ontology analysis tool. And these tools work fairly well for most of the analyses, but they still have some limitations that are important to be aware of. One of them being that it basically assumes that the gene or protein expression are independent, which we can safely assume is not exactly true, simply because if you have a set of proteins that are in the same protein complex, that are involved in the same biological processes, where often their expression will obviously be correlated. So this is the first step that there's one assumption which is violated in the first place in the type of data set that we are analyzing. Second limitation is that it doesn't consider gene or protein expression abundance. It's basically, are you among the significant proteins that are interesting for our study or are not? It's a binary feature. It's not how far you are in terms of abundance or in terms of confidence. So you sit here or here in the set of significant protein is not taken into account. The third limitation would be that it requires the establishment of thresholds that are chosen often by the user. For instance, here I've chosen a threshold for the abundance and for the confidence of the proteins to identify my set of interesting proteins I want to analyze. But the problem is that proteins that are sitting close to the border of these thresholds are treated very differently if they're on one side or the other of that threshold, although the differences are pretty minimal. And finally, most of these tools often yield very long lists of enriched GO terms that are often simply dumped in a paper and do not provide the information that people actually want because there's basically too much to filter. And so one of the tools that have been proposed a while back, now 10 years ago, to address some of these problems was Gsea, and I assume that most of you are probably familiar with the Gsea approach. So I will actually go quickly on that, but the idea behind Gsea was simply to statistically assess if a certain gene annotation or gene set, let's say, was significantly enriched towards the top or the bottom of a rank-ordered list of proteins or genes that are ranked ordered by their differential expression between two different phenotypic conditions. And so this approach is extremely popular and was extremely well received, but it has a few drawbacks and a few issues when we try to apply it to proteomics. The first one is that it requires two conditions. So you need to be able to contrast two different conditions in order to produce your ranked ordered list of genes or proteins. And this is not always the case in the proteomics data set. Quite often, people actually simply want to characterize a certain organism in terms of its proteome, and you only have a list of proteins that you've discovered, and there's no contrasting condition. And it's the same thing also when most of the time people are doing IPs. When they're trying to get the interaction of a given protein, we're simply providing a list of proteins, and we're not contrasting two conditions one against each other. The protein version of Gsea, which is called PSEA, and was published 5 years ago, the problem was that it does not handle label-based protein quantification, which is extremely popular these days. It also performs this very crude estimation on how protein abundance variation was captured, and proteomics results tend to be extremely variable, and reproducibility is always an issue. It would basically simply consider proteins variation based on their presence or their absence in a certain set of replicate. And the main problem that was an issue for us is that PSEA is not available as a user-friendly implementation, and that's a main limitation for obviously the researchers in the field. So our approach was inspired by Gsea and PSEA, and we named it PSEA-Quant. And basically our goal was to provide a statistical assessment of the overrepresentation of protein sets. So again, you can think of these about like coterms among proteins that are of high abundance and for which their abundance measurement have a low coefficient of variation in data sets that are composed of a single condition. So again, potentially IPs or simply the characterization of a given proteome. And so the idea behind PSEA-Quant is quite simple. Given the protein set, which here are the proteins in that protein set are annotated P1 to P7, we basically map these proteins in an enrichment score matrix based on their abundance in a set of replicates. So in this case, we've used spectral count as our measure of abundance, and their coefficient of variation among these set of replicates. So proteins that are highly abundant and that have a low coefficient of variation will end up in the top right of that enrichment score weight. And then in the end, once we're done mapping all of these proteins in our enrichment score weight matrix, we basically sum all their scores to obtain the protein enrichment score. Now the question that remains is, what is a good score for a set of proteins? Well, a good protein enrichment score that varies depending on the size of the protein set that you're analyzing and also the data set itself. So the strategy in order to evaluate what's a good protein enrichment score is basically to perform a statistical assessment, and we use a Monte Carlo sampling strategy in order to do so, where we basically sample at random in our data set another set of 7 proteins and compute the protein enrichment score, and repeat that procedure until we actually obtain an estimate of the distribution of the protein enrichment score for 7 proteins in our data set. And based on that, we can extrapolate, we can calculate a p-value, and therefore obtain the statistical assessment that we required in order to discriminate protein sets. In order to do the Monte Carlo sampling, there are several strategies that can be used. We can simply perform uniform sampling, where basically we would sample our 7 proteins in the example with a probability that is equal for all of them. This, however, assumes that all proteins have the same likelihood of being annotated in a protein set, and that is not exactly true, as we know there are literature biases people tend to study what people have studied before. So some proteins are a lot more characterized than other, and they are because they have already been characterized before. And so an alternative strategy, which is slightly more sophisticated, is to sample these X proteins, or 7 proteins in our example, with a probability which is roughly proportional to their respective number of occurrences in protein set. That basically takes into account for this literature bias I was referring to. We've also, for the sake of time, I will not go too much into the details of the methods that are implemented, but we've implemented a sampling space reduction model in order to model the protein expression dependencies that we might have in certain protein sets. And we obviously also perform a multiple hypothesis testing correction by deriving a false discovery rate for each of the p-values that are computed, because there are large numbers of protein sets that are tested. And that's obviously an important statistical requirement. So just to summarize the advantages that PSEA-Quant can have over other approaches, first of all, it can be applied on samples originating for a single condition, but it can also be applied in samples that involve more than one condition, and I'll show you how in a few seconds. It also does not require any arbitrary thresholds. A bit like Gsea and PSEA, it basically analyzes the entire data set and does not, it sees where the protein sits in that data set, and not require you to say that you want to see if there's an enrichment in a certain subset of interest in your data set. It models the likelihood of a protein to be annotated by a certain protein set, so that's that literature bias I was referring to. And also the part that I went over very quickly, where protein abundance correlations are modeled in our sampling procedure. So just as a quick example of the types of data set that PSEA-Quant can analyze, we had 3 biological replicates of a mud pit analysis of human bronchial epithelial cell line that was expressing a deletion of the amino acid 508 of CFTR. That deletion is known to be the main cause of cystic fibrosis, and so we perform the PSEA-Quant analysis on these 3 replicates, and we compared it to some of the state-of-the-art gene ontology analysis tools that are available out there. And interestingly enough, we found co-terms that are very much linked with cystic fibrosis that the classical gene ontology tool did not identify, simply because we're not asking the same statistical question. And so the role terms that were of interest involve cellular response to interleukin-4, which is a cytokine which is has an increased sensitivity in cystic fibrosis patient. Glucose and exose transport are heavily involved in disrupted in cystic fibrosis patient. Interestingly enough, we found GO-term involved with the protein folding and refolding. The mutation I was referring to on the protein CFTR involved a misfolding of CFTR, so again, that was very interesting. And a response to salt stress, if you're familiar with the CFTR mutation, you will know that the sodium channel of the cell is disrupted because of the misfolding of the CFTR protein. So all of these GO-terms were very much linked to CFTR and not identified by any Go analysis tools. Now, we can also contrast another condition. So if we have, for instance, 3 biological replicates in that case of the same cell line but expressing a wild-type CFTR, so a normally folded version of the CFTR protein, then we can contrast 2 conditions and see if certain GO-terms were significant in one of our analysis, for instance, where we had the CFTR mutant, and not in the wild type, so that we can see if certain pathways or biological processes are playing a role in the conditions that we're studying. And interestingly enough, we found more or less the same GO-terms that are related to CFTR to be significant in the data set where we have the mutant CFTR and versus the data set with the wild type version of CFTR. Now, some of the future work for PSEA-Quant involved protein set clustering. There was in the protein sets that I showed you before, there was some manual curation to remove redundant Go terms, and that's a big problem often, is that most of these terms are quite overlapping, if not exactly the same, and there needs to be work for PSEA-Quant in order to cluster these results in a meaningful fashion so that the user can browse through the result in a more efficient manner. And there's also the implementation of a larger variety of features besides the abundance and the coefficient of variation in order to perform the enrichment score calculations. So for instance, peak intensities could be used, or simply coefficient of variation, depending on what are the goals and what are the statistical questions that the user wants to ask. So I guess in the quick conclusion, I've shown you that PSEA-Quant can identify protein sets that are significantly enriched among high abundance proteins that are for which the enrichment are well reproduced. PSEA-Quant does allow a PSEA type analysis on single condition data set, but can also be used for two conditions. PSEA-Quant can perform a sampling procedure that models both the expression dependency and the literature biases in terms of protein set annotations. And I should mention, and I think that's very important again, and I hope I was clear on that point, that PSEA-Quant is complementary to classic gene ontology analysis. It's not necessarily better, it's just asking a different question. We're looking at the enrichment among abundant proteins in the data set, whereas gene ontology analysis are simply looking at the presence or absence in a certain set of proteins, if these presents are enriched or not. So they can yield complementary results that are interesting and might yield more information about a certain data set. And PSEA-Quant can help characterize data set and obviously formulate research hypotheses based on the results. Just before I go just on a quick demo on how the tool works, I want to acknowledge obviously my supervisor John Yates and Naveen and Daniel for producing the data, and all of the research team, the bioinformatics research team in the Yates lab that you've had the chance to have a talk from over the last few weeks. So Salvador and Robin, and obviously all the PSEA-Quant users. So PSEA-Quant was released officially in October of last year, and we already have more than 50 labs that are using it. So we're quite happy, and we hopefully think that the number of users will augment over the following years, and obviously the funding that produced the tool. So PSEA-Quant is a web-based tool. It is a fairly simple web interface, which we think is fairly user-friendly. And I'm just going to quickly switch over to give you a chance to see what it looks in action. There you go. So basically, it's a simple web form. There are simple input files that are accessible if you simply want to try the tool and make sure your input is properly formatted. But the format is pretty simple. So something I did not went over in the presentation for the sake of time is that yes, spectral count in terms of protein abundances can be used as an input, but also abundance ratios. So if you are using some sort of label experiment, TMT, iTrack, SciLac, N15, basically all of them, whatever gives you a set of ratio in terms of abundance can be used, so that helps contrasting two conditions in one single run. You can also use ratios of spectral counts if you have two conditions, for instance. For now, it supports 3 organisms: Homo sapiens, mouse, and rat. We are about to include 2 more. That includes the Drosophila and yeast. Hopefully, over time, we'll include more and more of these. Then you can input your data set, and it's a simple comma-separated vector format where you have the protein name or the UniProt ID of a protein. Protein symbol, sorry, and the UniProt ID of a protein, and then the abundance value, whether they're spectral count or abundance ratios. And it can support any number of replicates. and then you can choose the file, which I've already done here. Then you can select the number of samplings for the Monte Carlo sampling procedure. The less sampling you do, the faster the analysis will be, the less accurate the p-values will be. The more sampling you do, the more accurate the p-value will be, but the longer the execution will be. So we recommend 10,000. We believe we get reasonably good estimates of the p-values based on our analysis with 10,000, and so that's a good trade-off in terms of running time and accuracy of the results. And then you can choose the annotation databases. We're supporting both the gene ontology database and the MSigDB, the molecular signature database that was in the original Gsea publication. However, this is a human-centric database, so this can only be applied on Homo sapiens. And then the sampling procedure I was referring to quickly, if you want to assume that the protein expression abundant is dependent or not, and we have this little factor that we recommend to put around 0.5, so all the default values are the recommended values. And if you have more questions about this, I can go a little bit more into the details after. And finally, the literature is in terms of the weighted sampling for the Monte Carlo sampling procedure, which we recommend to perform, is already selected here. And finally, the final step is to enter your email address, because unfortunately, since the method requires a Monte Carlo sampling analysis, we're talking about a fairly long running time. It's not something that is done in 2 seconds, but it's more in terms of minutes and sometimes a few hours based on the load on the server and the number of the types of parameters that are chosen. And so basically you can click submit, and what happens, why we ask for that email, is that once the analysis will be completed, an email will be sent to the address of the user containing the path of where the file is actually been, the output file is located. And so for now, the file will obviously not be ready because of the time it will take, but in the end you receive an email in order to see your output file, so that's actually pretty convenient. I think that's it. I guess thank you, Vincent, and everybody for setting up the presentation and listening to my talk, and I'll be happy to answer your questions.
Thank you, Matthew.
So this is Andrew Sue. I have a question here. Actually, first, a very quick suggestion. It'd be actually really cool to put you have sample input files on your website. It'd be nice to put sample output files corresponding to that. But my question is this, right? So I want to go back to this idea of doing a single condition analysis, right? So you have this background, weighted sampling according to how many gene sets or protein sets a given protein occurs in. But does that account for, I mean, isn't there also a bias in terms of just the experimental sampling? In terms of, certain proteins have more peptides that will fly versus not. Is that a confounder that you need to take into account?
Yes, that's a very good point. So I guess in the first place, I took good note of your comment. I agree that's a good idea to put the sample output file, and I will do that as soon as possible. Your second point is also very true. It is a confounder. There are proteins that are simply more likely to be abundant than others because they fly better than others. However, in the sampling procedure, when we perform the FDR analysis that I've very briefly introduced, we do perform a shuffling of the annotations, and so therefore these very abundant proteins will get a new set of annotations, and therefore we basically see if a p-value with that given protein happens fairly often, and we therefore readjust the p-values based on that shuffling of annotations, and that's therefore taken into account as this this confounding factor is taken into account with the FDR analysis.
Got it. Thanks. Cool work.
Thanks.
Are there any other questions for Matthew? Okay. Thank you, Matthew, for that presentation.
Thank you, guys.
Yeah. So we'll move on to our next presentation. So I'll be talking about some of the updates that we have from COPaKB. Yeah. Presentation. All right. So I'm Vincent Key. I'm the BD2K DSR project manager and also a COPaKB developer. And I'll give a brief update on COPaKB. So I'll first go over some of the existing features that we have. So currently COPaKB features high quality cardiac proteomic data of 10 modules spanning multiple species and organelles within each species. And we support 4 different query types, including protein identifier query using the UniProt ID or the gene name, peptide sequence query, spectrum precursor query, and also MS data file query supporting MDML and DTA formats. And we also welcome researchers to share their mass spec data to be incorporated in our knowledge base. Some of the new features that we're looking to develop is, so we have the first 2, which is protein batch query and cardiovascular disease query. And that's still in the process of defining the exact specifications, but we have an overall idea of what we're trying to create. And for the last 2 features, which is the Gene Wiki link and the COPaKB API, we're currently in the development process. So first, I'll go over the protein batch query. So the idea here is that our user has a list of proteins, and he wants to see what cardiovascular diseases are associated with the particular list of proteins. So the user submits a list and clicks Analyze, and COPaKB will give a table view of the cardiovascular diseases mapped to the proteins. And diseases are mapped using PubMed and online Mendelian inheritance in man, or OMIM. And this the basic idea of this feature is users can identify which proteins are associated with which disease. And in the future, we may include a statistical value, like false discovery rate, to show the significance of the results. And also, we're also thinking about creating a cardiovascular gene ontology analysis. So for those who aren't familiar with gene ontology, it is a vocabulary that is established standard for the functional annotation of gene products. And the Cardiovascular Gene Ontology Initiative is led by University College London. And currently there are over 4,000 cardiovascular associated genes that have been identified as targets for annotation with Go terms. So same here, a user submits a list of proteins, and he'll be able to see which proteins are associated with which Go term. And as you can see from the table, each Go term is mapped to one or multiple proteins. And another feature that we're hoping to develop is a cardiovascular disease query. So we currently have a mapping of diseases to proteins that are curated from PubMed and OMIM. Now this information is currently in our database and is used for our protein query search, in which we provide disease relevance when a user searches for a protein. And now we're hoping to make the disease searchable. So for example, a user can look up a cardiovascular disease and search which proteins are associated with the disease. So this feature, we're hoping, will allow cardiologists or citizen scientists who are not familiar with proteins to perform this search to learn the role and function of the protein in the disease and medicine. So we'll be giving a list of proteins, and then a GeneWiki link will be provided to give them more information about that protein or gene. And also our lab has been working closely with Dr. Su's team on the GeneWiki project. So we're focused on populating wiki pages of proteins or genes relating to the mitochondrial proteome. And we have assessed 642 proteins or gene pages for overall completion, content, and organization. And in addition, we'll be embedding these GeneWiki articles into our website that will provide users with more information about that protein or gene. So a quick example of what it will look like is, so a user will go to our page, and when they want to search for a protein, you'll get some basic information about that protein, and also a tab will be provided below with the Wikipedia page embedded in the tab, so they can quickly access more information about a particular gene or protein. And then we have our API also that we're currently developing. So we have 5 different functionalities, but we're focusing on the first 3. So the protein annotation service, the peptide annotation service, and also the MZML analysis service. And then later, after we complete that, we'll be adding the MS data file DTA search and also the spectrum precursor search. So the first one that I'll go over is the peptide annotation retrieval. And that basically allows you to search a peptide, and it will return all the relevant spectra that's in our knowledge base, and also the proteins that contain that peptide. So here, all you have to do is put in the URL and the peptide sequence, and by default, it'll show you all the relevant information associated with the peptide. And then you can also provide filters. So if you just want the proteins, then you can just put /proteins or just spectra info. So here's an example of what that JSON document would look like when you search peptide sequence. And another feature for our API is so another feature is the protein search. So with this, you provide a UniProt ID, and you'll get all the relevant information about that protein. So that includes sequence information, all the contained peptides that we have in our knowledge base, cardiovascular disease relevance, the expression profile, and also a list of interactions with other proteins. So here we have another example of the JSON document. So this is the default document that you'll get when you search protein. And you can also provide filters based on what you need. And our third feature that we're hoping to develop for the API is the MZML analysis retrieval. So I'll quickly go over the three-step process to upload and retrieve the analysis. So first, you'll upload a MZML file onto the website. We're currently looking into finding alternatives to upload the file. So currently we use a .NET framework called Neat Upload, but we're looking for faster alternatives. Maybe looking into Aspera or another transfer protocol. So once you do that, you'll receive a token, and then you'll be able to retrieve the analysis using our API. So first, you'll go to our website and then submit your MZML file, and you'll need to provide an email address, and also you can upload your file here, and then click Analyze. And then after clicking Analyze, you'll get a task ID that appears after you click Upload and Search. And we're working to replace this task ID with a randomized token, so that it's not just a consecutive number like 15062, and this will be for security purposes. So once the analysis is finished, you'll receive an email saying that your analysis is finished, and you'll receive a token. And that so with that token, you can access your report through API. And by default, you'll receive information about, or all the information about the proteins that were found in your from your experiment, and also all the peptides that were found that were part of that protein. So with that report, you can also filter out by getting just the proteins, or you can also search within the proteins the peptides that were found in, from your experiment. Here's a quick example of the Json document that you get. So you get the list of proteins here, and then on the right side you see the list of peptides from your experiment. So overall, so our current progress, we've developed a prototype for the API, but we're looking to change the API specifications to be more compliant for the expression interface, and we're also restructuring the backend for our API to speed up our performance. So thank you for listening. I'll be happy to take any questions that you may have.
Vincent, I would have two questions. The first one, I couldn't help but see how useful that your analysis would be to be included with the PSEA-Quant. So I was wondering if it was possible for you to produce some sort of mapping file from the proteins that have any of the OMIM annotations and the other databases that you're querying in terms of their association with cardiovascular diseases, some sort of output mapping file that could be used for somebody that wants to do some other analysis on them.
Did you get the question? Yeah. Wait, could you repeat the question?
Yes. So I was wondering if you had some sort of association file where you would have all the proteins that are annotated with cardiovascular disease and basically all the diseases they are associated with based on all the databases that you're and the resources that you're querying when somebody outputs a search for a given list of protein.
So it's PayPay. Maybe I could help to answer that question. At the moment, most of COPaKB protein data sets are organized based on organelle functions.
Okay.
And obviously, if there is a disease, there will be dysfunction of that organelle. And so there have been some efforts to put both a healthy organelle as well as a diseased organelle protein modifications to cluster and aggregate them. But we're not there yet. It's going to take some efforts, with collaboration with PRIDE, where it collects multiple diseased proteome data sets from the past few years' publication. And we probably will get there in the near future, but it's a very important task that we have on our priority list.
I see. Okay. Thank you. And if I can, I have a second question. When a user submits an MZML file, I guess, are there some sort of parameters that should be also given with the MZML file regarding the search in order to identify the peptides that are present in there?
Yes, there are. Just a second. Yeah, they're on the screen. You can see there's some search parameters.
Okay.
Yeah.
I was just curious. Okay, that's great. Thank you very much.
Are there any other questions? Okay, thank you all for listening. So just a quick update. So we're just about finished with our first round of presentations. We still have one more, maybe next week, but we're basically finished. So next week, we'll have more information about what our next round of presentations will consist of. Maybe some updates regarding all the tools, or if anyone wants, they can request more information about a particular tool, and we'll try to schedule presentations for those tools. All right, but thank you all for attending this week's conference. You all have a good day.
Thank you.
I just have one more thing. If any one of you who has a colleague and doing fabulous data science or any tool development that you would like to recommend to give a talk on our Friday technical calls, please just send the information to either myself, Andrew, or Henning, and Carol or Mary. Will have a discussion. We'll recommend them to Vincent and Brian, and they will help us to collaborate with other people in that regard. Thank you.
Yeah, thanks. Sounds good. All right, thank you.